So our next topic is muscle metabolism, and this is where muscles get energy for contraction, because ATP is the only energy source used directly for contractile activity. And we don't really store a lot of ATP in our cells, specifically not our muscle cells, only 4 to 6 seconds worth, so we have to constantly generate ATP. So as soon as the available stores of ATP are hydrolyzed or broken down to release energy from their terminal phosphate, they're regenerated by the following processes: direct phosphorylation, anaerobic glycolysis, and aerobic respiration. I want to point out the terms anaerobic and aerobic. Anaerobic means without oxygen, and aerobic means with oxygen. Also, the process of glycolysis occurs during aerobic respiration, but aerobic respiration just includes more than just glycolysis, which is what anaerobic glycolysis stands for. So here are the 3 parts of muscle metabolism. So for direct phosphorylation, we're going to utilize creatine phosphate and ADP to generate creatine and ATP. The energy source for this step is creatine phosphate, and we're not going to use any oxygen, and we'll create 1 ATP molecule per 1 creatine phosphate molecule. We're also going to see that this happens relatively quickly, and it's facilitated by the use of the enzyme creatine kinase that makes sure this reaction happens quickly enough to create ATP that can sustain muscle metabolism for a short period of time. So what's good about direct phosphorylation? Well, it uses a readily available resource, creatine phosphate, to make energy. It's quick, it only takes 15 seconds. But it doesn't create a lot of ATP. It only creates 1 ATP molecule per creatine phosphate. But it doesn't require oxygen and just this really easy to come by creatine phosphate, so it's not bad. What about anaerobic glycolysis? Well, anaerobic glycolysis also doesn't require oxygen. We actually get 2 ATP per glucose because glucose is our reactant or our energy source. We basically break down glucose, which is a 6-carbon structure, into 2 3-carbon structures called pyruvate. And during that process, you create 2 ATP. And since oxygen is not used, that's not bad. Glucose is pretty readily available. We get it from the food we eat. This process could also use fat if it wanted to. And it's pretty quick. It only takes 30 to 40 seconds. But again, we're only getting 2 ATP for all this work. So creatine phosphate, glucose, these processes, direct phosphorylation, and anaerobic glycolysis, they seem useful for quick energy, but not a lot of it. What muscle cells and other cells use the most is aerobic respiration, or what's called the aerobic mechanism. Aerobic cellular respiration uses oxygen, and glycolysis still takes place, so we break down glucose into pyruvate, but then the pyruvate enters the mitochondria, and the mitochondria uses the pyruvate in order to generate ATP. We also generate some carbon dioxide, which you breathe out as a waste product, and water, which is also a byproduct. But our net gain of glucose is 32, sometimes 32 to 34 ATP. So why don't we just use this mechanism? It creates tons of energy compared to direct phosphorylation or the anaerobic mechanism. The reason or negative to aerobic cellular respiration is it takes hours. Now cells still use this because it creates so much ATP, but if we need quick energy, if oxygen cannot get to muscle cells, say you're exercising and your knees are bent because you're running, so there's not readily available oxygen to the muscles of your calf immediately or always, then there'll be a little bit of anaerobic or direct phosphorylation that occurs. So it's great that cells have all these options, but the option called aerobic cellular respiration produces the most ATP, and even though it takes hours, it's still the most beneficial for creating readily available and usable energy that lasts longer. So let's look at each one separately again. So muscle cells can actually store 2 to 3 times more CP, which is creatine phosphate, than ATP, and that was our energy source for direct phosphorylation. So if we need more ATP, once we've used our stores of ATP, direct phosphorylation is a great way to get it. But even though we store 2 to 3 times the amount of creatine phosphate then ATP in muscle cells, it can be used up really quickly because it takes a complete creatine phosphate molecule to make 1 ATP. So we don't get really much for the use of our creatine phosphate. So we would use pretty much direct phosphorylation to generate ATP while the metabolic pathways adjust to the sudden higher demands. So if you go from sitting down to suddenly running, then we're going to use creatine phosphate first before your muscles are prepared to use the aerobic mechanism of cellular respiration. And that works great because we can get ATP again really quickly, 15 seconds. We don't get much for our use of creatine phosphate as the energy source. Then, when muscle contraction activity reaches about 70% maximum, if oxygen delivery becomes impaired, we'll use anaerobic glycolysis instead of creatine phosphate via direct phosphorylation. The problem with anaerobic glycolysis is that if we can't recycle the lactic acid produced from the pyruvate that glycolysis is turned into, then that ends up in the muscle itself and can cause muscle soreness. But if muscles are bulging because they are being contracted, therefore compressing blood vessels, and oxygen is not to be had, anaerobic glycolysis is a great example. But again, we don't get much ATP. Even though it's quick, it's a little bit longer than direct phosphorylation, and we only get 1 ATP made per glucose molecule. So the anaerobic glycolysis mechanism can form lactic acid, and that can cause muscle soreness. But we also see that anaerobic glycolysis is the initial phase of the aerobic pathway. So even though the initial phase of the aerobic pathway proceeds without oxygen, and itself could make 2 ATP per glucose molecule, and it's short, it's still better if we use anaerobic glycolysis as the priming event for cellular respiration. And that mainly is because of that lactic acid. If it doesn't diffuse into the bloodstream, get picked up by the liver, the kidneys, and the heart, and converted back into pyruvic acid by the liver, then it ends up in the muscles and can be partially responsible for why they're sore. Other parts or things that are responsible for the soreness of muscles is fatigue. So really, we like the aerobic pathway that is primed by the anaerobic glycolysis pathway and then continues with the use of the mitochondria, and this pathway does require oxygen. This is the way that we make 95% of the ATP used for muscle activity, and yes, it does include that anaerobic priming step, but the full breakdown of glucose occurs in the mitochondria. But it takes hours, but we get 32 to 34 net ATP per glucose molecule. And then carbon dioxide and water are byproducts or waste products. So here we have again a muscle metabolism. So there is direct phosphorylation, which uses creatine phosphate as its energy source, anaerobic glycolysis that uses glucose as its energy source, and the aerobic respiration method, which uses glucose mainly as its energy source, but could also use pyruvic acid, fatty acids, amino acids, and pretty much a lot of different types of biological molecules once broken down. Now, creatine phosphate and glucose are great because they can be utilized to create ATP, but direct phosphorylation only creates 1 ATP per creatine phosphate, whereas per glucose breakdown during the anaerobic mechanism, we get 2 ATP per glucose. Both of these are quick, 15 versus 30 to 40 seconds, but we get the most ATP using the aerobic mechanism, so that's why most of the ATP that muscle cells use for metabolism is made using aerobic respiration. So if you were asked which of the following methods produces the most ATP for the use of contraction, or for the use of energy for contraction, you would say the aerobic mechanism or aerobic cellular respiration. So speaking of lactic acid and muscle fatigue just occurs when the muscle organ is in a state of physiological inability to contract. So if we can't keep pace with the ATP needed for contraction, so ATP production fails to keep pace with ATP use, muscle fatigue can occur. With a deficit of ATP, that can result in the muscle organ contracting irregularly. It can cause lactic acid to accumulate in the muscle and ionic imbalances present in the muscle. And pretty much all of that would have to be corrected in order for fatigue to end. So for a muscle to return to its resting state, it has to pay the oxygen debt, which is now called the excess post-exercise oxygen consumption. Personally, I prefer oxygen debt. It's simple and it makes sense. This is what the muscle must pay or replenish in order to be able to contract again. That includes replenishes oxygen reserves. Make sure that myoglobin is holding on to at least some stored oxygen. Make sure that accumulated lactic acid be converted to pyruvic acid and removed from the muscle itself. Make sure glycogen stores are replaced. So there are some glycogen or glucose that is stored in muscle cells, and glucose again is used during the aerobic respiration stage because glucose is broken down completely in the mitochondria, and that aerobic respiration is what produces the most ATP. And speaking of ATP and creatine phosphate reserves, even though it's just a little bit, must be resynthesized in order for a muscle to return to a resting state. So essentially, if a muscle is fatigued, it must pay the oxygen debt in order to contract again. Let's also talk about the force of muscle contraction, and all of these should make sense. If more muscle fibers contract, then you have a stronger contraction. If the muscle size is larger, meaning it's a bulkier muscle, it has greater strength. If you increase the frequency of action potential stimulation, so basically have a more rapidly stimulated muscle, we're going to have greater force that the muscle can exert. And if a muscle's contraction occurs when the fibers are at 80 to 120 of their normal resting length, you're going to have more force of that contraction. So basically, as long as muscle contraction happens when there is a good degree of overlap between actin and myosin and the myofibrils, you'll have the ability for the muscle to contract better than if it was overstretched or too shortened. So these are all the things that are going to affect muscle contraction: the number of muscle fibers contracting, the relative size of the muscle, the frequency of stimulation, and the degree of muscle stretch. There's also muscle fiber types. So these are just functional characteristics of the muscle fiber types, and then we'll look at the actual types. They are going to be functionally classified as whether or not they break down ATP quickly or slowly. So that's the speed of contraction, which is determined by the speed in which ATPases, which are enzymes that split ATP, break down ATP. You can either break them down slow or fast, and we'll just use those terms to describe the muscle fibers. You can also use the ATP forming pathways to describe the fibers. If they're oxidative fibers, they use the aerobic pathways, also sometimes called type 1. If they are glycolytic fibers, then they use the anaerobic pathways, specifically anaerobic glycolysis, which occurs without oxygen. So these are the two criteria we're going to use to define and categorize the muscle fiber types. And our muscle fiber types, therefore, are slow oxidative, fast oxidative, and fast glycolytic fibers. The slow oxidative fibers contract slowly, whereas the fast oxidative and fast glycolytic fibers contract quickly because of the quick-acting ATPases. The fast and slow oxidative fibers both depend on oxygen because they use the oxidative or aerobic respiration pathways. Slow oxidative fibers are fatigue resistant, so we see marathon runners have more skeletal muscle fibers converted into this type, whereas the fast glycolytic fibers use anaerobic glycolysis, and even though they have fast myosin ATPases, they're easily fatigued, so sprinters have more skeletal muscle fibers converted into this type. Whereas the fast oxidative fibers, they have more moderate resistance to fatigue, but they're not fatigue resistance, but they're not as easily fatigued as fast glycolytic fibers. So we don't necessarily see marathon runners have more of this type or sprinters have more of this type. It's kind of an intermediate type of muscle fiber that you may find in marathon runners, sprinters, and just that average everyday person. And last but not least, let's talk about some differences of smooth muscle compared to skeletal muscle. We know smooth muscle cells are spindle-shaped fibers. They have a similar diameter to skeletal muscle fibers, but sometimes much smaller, 2 to 10 micrometers in length, and only 700 micrometers around. And they lack the connective tissue sheaths that we've been talking about, like the epimysium and the perimysium, but they do still have a very fine endomysium. Usually we're going to see that in the walls of hollow organs, that smooth muscle is organized into two layers, one layer with cells going longitudinally, and the other layer with cells going in a circular manner. And that means that the two layers are opposed, or that the fibers are perpendicular to each other. Keep in mind, smooth muscle is found in the walls of hollow organs. We find it throughout the body in all of these hollow organs except for the heart, which is made up of cardiac muscle. And we have pretty much the same contractile mechanism as skeletal muscle with a couple of changes. So first, let's look at its anatomy. We know it's in the walls of hollow organs like digestive organs, the urinary bladder as well. And here are two layers. You can see that some of the cells look like they'd be coming at you, that you've taken a cross section, whereas the others are pointing kind of away from you to the left and right. So one layer would be longitudinal, the other layer would be circular. The circular and longitudinal layers would contract at different times to allow movement through a passageway. So in those hollow organs, the longitudinal layers of smooth muscle would contract, causing the hollow organ to dilate, whereas if the circular layer contracts, the organ lumen is going to constrict and elongate. So this back and forth dilation and constriction and then elongation is able to squeeze foodstuffs like chyme or waste through hollow organs. This back and forth dilation and constriction that causes elongation is called peristalsis, and is that alternating contraction and relaxation pattern that we're seeing in smooth muscle that's able to allow it to mix and squeeze substances through the lumen of hollow organs. Smooth muscle is also innervated, but it receives its innervation from the autonomic division of the nervous system as opposed to the somatic nervous system, which provides the innervation of skeletal muscle. We also know that the hormones that control the contraction of smooth muscle are neurotransmitters like norepinephrine as opposed to acetylcholine. But there's still local chemical changes, things like excess carbon dioxide or low pH that can also cause the contraction of smooth muscle cells. It doesn't have to be norepinephrine. We don't have any neuromuscular junctions in smooth muscle. Instead, there's varicosities or bulbous swellings at the end of axons. The varicosities release neurotransmitters just like the axon terminals of the motor neuron axons for skeletal muscle do, but there's a much wider synaptic cleft, so we have diffuse junctions as opposed to neuromuscular junctions. So here are the varicosities. So varicosities are bulbous swellings at the end of axons that are going to be associated with the innervation of smooth muscle. You're not going to have a very well-developed sarcoplasmic reticulum. As we saw in skeletal muscle and smooth muscle, we're not going to have that specific pattern with T-tubules abutting this terminal cisternae of the sarcoplasmic reticulum, because the T-tubules are actually absent in our smooth muscle. Instead, you have on the outside or the plasma membranes of smooth muscle cells a pouch-like infolding called a calveoli, where there is extracellular calcium. And this extracellular calcium is going to be part of the trigger for the release of calcium from the sarcoplasmic reticulum. And because there is not a regular arrangement of actin and myosin in smooth muscle, there are no visible striations and no sarcomeres, because, again, sarcomeres are units of myofibrils that have regular arrangements of actin and myosin. Keep in mind that actin and myosin are still present. We just have these minor variations, no sarcomeres, therefore no striations. But the ratio of myosin to actin is going to be much lower than we saw in skeletal muscle. So still present, just not in as much of abundance. The thick filaments and thin filaments are still going to have to form a cross bridge in order to cause contraction, but their arrangement is diagonal, meaning that when smooth muscle contracts, it contracts in a corkscrew manner. And instead of having Z discs, because that would be the end of sarcomeres since we don't have sarcomeres, there are areas where the actin and myosin attach to the smooth muscle at dense bodies. So these are just non-contractile intermediate filament bundles where the actin and myosin attach together. And there's no troponin complex. So here we have the contraction of smooth muscle. We see a relaxed smooth muscle up top, contracted at the bottom. You see that the actin and myosin is attached at intermediate filament bundles called the dense bodies. We see the spaces outside of the plasma membrane in the top image. Those are calveoli. Those calveoli are where we would have extracellular calcium being sequestered in order for it to enter when we need it to cause the release of calcium from the sarcoplasmic reticulum. Down at the bottom, we see the contracted smooth muscle cell. We can see the slight corkscrew motion that it takes when it contracts as well. And whole sheets of smooth muscle are actually going to contract at once. There's synchronized contraction. They exhibit slower contraction, about 30 times slower, but it's still very synchronized. So they contract in unison because the cells are connected via gap junctions, and gap junctions allow the electrical signal to travel from cell to cell. Therefore, once initiated, action potentials are very easily transmitted from smooth muscle fiber to smooth muscle fiber. And that occurs again in the same sheet of muscle. Some smooth muscle cells have pacemakers like cardiac muscle cells, and they would set the contractile pace for the whole sheet of muscle, for example, in the wall of a hollow organ. So planning yourself excitatory and depolarized even without external stimuli, even though smooth muscle is externally innervated. The contraction mechanism is really similar to skeletal muscle, but some changes. Again, we have that extracellular calcium in the calveoli. That's a little bit different. But the final trigger for contraction is still a rise in intracellular calcium. Just some of it enters from the outside. And then that is the trigger for the rest of the calcium release from the sarcoplasmic reticulum. And that initiates the sliding filament model of contraction between actin and myosin. But in this case, since there's no troponin, tropomyosin complex, calcium binds instead to calmodulin and activates that. And then the calmodulin is going to activate kinase enzymes. Kinase enzymes are going to take a phosphate from ATP, thereby releasing energy from ATP to form the myosin cross bridges. So ATP is still required to activate the myosin head and to allow it to bind to actin. And then the phosphorylated cross bridges are able to interact, produce that sliding filament mechanism where we see greater overlap of actin and myosin, therefore contraction of the smooth muscle. And then it'll relax when phosphorylation decreases and the intracellular calcium levels drop. Some of that calcium will be pumped out of the cell and some of it back into the sarcoplasmic reticulum. And that is it for Chapter 9. Make sure to review all those A&P Flex videos. I will not specifically take information out of those videos because all the information is available in your PowerPoints and your lecture textbook. But the videos are very helpful for visualization of this complex anatomy, specifically the microscopic anatomy that has to do with the steps for skeletal muscle contraction, which again are the neuromuscular junction, excitation-contraction coupling, and the cross-bridge cycle. So make sure to read your textbook and review all parts of the video lecture.